Hi everyone, and welcome to our first screencast about enzymes. By the end of this unit, you'll need to know and understand all of these terms: metabolism, enzyme, substrate, know what a coenzyme is, and what the energy of activation is. You'll also need to know what the thyroid gland does and how the hormone that it makes, thyroxine, is related to metabolism. We'll also be learning why enzymes are very much like a lock and key when they interact with substrates. You have to know how vitamins are related to enzymes and differentiate between what an enzyme and a coenzyme do when they are involved in reactions. And then take the information that you know about proteins and explain how the activity of enzymes can be influenced by different things like pH and temperature, concentration. You also need to know lots of vocab for this section, so make sure you can check off all of those as, yes, I definitely know those definitions. So what is an enzyme? An enzyme is a catalyst in a living thing, and it helps to speed up reactions. And they don't get used up in a reaction, so they can be used over and over and over and over again. Enzymes, we know, are proteins. So some proteins are enzymes. They're reusable, and because they are reusable and they can work over and over again, you only need them in very low concentrations. You don't need very many of the enzymes to speed up the reaction rate of something that's happening inside your body. So you don't need much. Some examples of enzymes are amylase. Amylase is an enzyme that cuts up starch and turns it into sugars. This happens in your mouth. Lipase is an enzyme that cuts up lipids and turns them into fatty acids and glycerol. This happens in your intestines. And proteases are all the different types of enzymes that chop up proteins and turn them into amino acids. This happens in your stomach and in your intestines. You'll notice that enzymes tend to end in ASE. Anything that you see, A's, amylase, lipase, is probably an enzyme. So we've already talked about some enzymes. We've talked about helicase that chops up the hydrogen bonds in DNA. We've talked about DNA polymerase that forms the sugar phosphate backbone during DNA replication. We've talked about RNA polymerase, which forms the backbone when you're making messenger RNA. We've also talked about peptidyltransferase, which is the enzyme that forms that peptide bond between two amino acids at the ribosome. So what an enzyme does, the way that it speeds up a reaction is it allows reactions to happen at lower temperatures than they would normally be able to occur. So why don't we just, instead of making enzymes, which takes a lot of energy, why not just crank up the heat, make it real hot inside our bodies? Well, we can't do that, and you should already know and remember that denaturation happens to proteins when they're too hot. They lose their shape and they can't do their job anymore. So what enzymes do is they help to either split apart or join together certain reactants in our body, certain substrates. When you split something apart, for example, splitting apart starch into sugars, that's called hydrolysis. And when you join things together, for example, joining amino acids together to make a protein, that's dehydration synthesis. And that's what enzymes do. They do hydrolysis and they do dehydration synthesis inside our bodies. And the substrate is the thing that is being changed, the reactant. So if you do take starch and you turn it into maltose with the enzyme amylase, maltose is a disaccharide, then the starch is reactant, the starch is the substrate. Maltose, what the starch gets chopped up into, is the product. So enzymes turn substrates into products. And there's a few steps involved in this. One of the steps is you'll end up with an ES complex, an enzyme substrate complex, when the enzyme and the substrate are fused together and the enzyme is putting stress on the substrate to either break it apart or join it together. So here we see two substrates coming in to the active site of the enzyme, and the enzyme causes stress and ultimately synthesizes, dehydration synthesis, joins those substrates until they become products. So an enzyme, we know proteins from our study of proteins that enzymes are three-dimensional globular proteins. They have a very specific shape. This is the tertiary structure of a protein that I'm talking about. And you'll notice right here in this section of the protein, that is called the active site. And the active site is where the magic happens. This is where the substrate will join the enzyme. Enzymes have really special grooves or pockets, and that makes up the active site. And inside this active site, there's functional groups, R groups, that have special qualities that help the enzyme either break apart or join together substrates. So they have R groups that can interact with the substrate. So these are called active sites, and it's where the substrate attaches. So if you look at this picture, this is kind of a cartoon picture. Here's the active site here. And enzymes are very specific, and they only fit one type of substrate. So if you look here, substrate A and substrate B will join the active site of this special enzyme. When they're joined, they're called an ES complex, and then the enzyme will in some way change those substrates to form a product. In this case, it synthesized them. It formed a bond between them. This lower picture here is more realistic because we don't really have proteins that look like cartoon shapes, but here you'll see there's a special little groove, a special little indent, and that is the active site of an enzyme. So we call the interaction between the active site and the enzyme, because they're such specific shapes, we can refer to it as the lock and key model. They're very specific. So just like you've got one key type for your front door, that's the same as an enzyme. An enzyme has a certain shape, and that shape will only fit one specific type of substrate. And they fit perfectly together like a lock and a key. So if we use our analogy, the enzyme would be the lock, and the substrates would be the key. When the enzyme and substrate come together, the shape of the enzyme actually changes. As soon as they touch each other, the shape of the enzyme changes and it becomes more reactive. So you see here in this beginning, when they're not touching, the black enzyme shape isn't perfectly fit to the substrate. But as soon as they make contact, the enzyme will shift and move until it fits perfectly the enzyme. And it keeps moving until it stresses the substrate in some way. This is called induced fit, because as soon as they touch each other, the enzyme will fit exactly properly onto the substrate. And there's two parts of the enzyme. One part of the enzyme is called the apoenzyme. That's the part that we've been talking about so far. The other part is called the coenzyme. It's not a protein. It's called either a cofactor or a coenzyme, and it's usually a vitamin. So it's not a protein at all, and what it does is it will join either the active site or a different site, and it helps the substrate fit into the enzyme exactly perfectly right. So and when these join together, when the apoenzyme and the coenzyme join together, it basically activates, makes the enzyme ready to do its job. The other type of coenzyme is the type that joins in the allosteric site. This is a site opposite on the other side from the active site. And when the coenzyme joins there, coenzyme will change the shape of the active site, and it'll allow the substrate to fit properly. You'll see here it doesn't fit properly, but when that coenzyme joins, the shape of the enzyme's active site will fit the substrate perfectly. This is just another example of the induced fit. When everything is where it's supposed to be and the substrate just touches the active site and the coenzyme is in place, then you've got an induced fit. So coenzymes, like I said, are usually vitamins, so we get those from our food, we get them from our supplements. But if you're eating a balanced diet, you don't really need to take vitamin pills. But that's not true for all vitamins, and that's why they started putting folic acid, which you usually get from dark green vegetables like spinach. But in 1999, the states realized that people weren't eating enough spinach, and so they just started putting folic acid into our flour. So you get it from eating bread and pizza. So now we need to talk about anabolism and catabolism. And we know that the active site of an enzyme isn't exactly a perfect fit. And like I said, this causes stress in the substrate. And that stress will either cause one of two things. It will either cause the substrate to break apart, and that's what you get with a hydrolysis reaction. We know you need to add water to make this happen, and then hydrolysis occurs. So you're breaking a big molecule, a polymer, into a smaller molecule, a monomer. And another word for this is catabolism. So just picture a big cat using its claws to rip apart a molecule, a big molecule, into a smaller molecule, as you can see in this animation. So one substrate becoming two products. Another example of that is sucrose. So if you've got some table sugar, sucrose, and you add some enzyme, you add the sucrase enzyme, you can see it ends in ase, then it will take sucrose and you'll add some water because we're doing hydrolysis, and the enzyme sucrase will cause enough stress on the sucrose to break it apart into glucose and fructose. This is an example of catabolism, ripping a molecule apart into two products. This happens regularly when we're digesting our food. The second thing that an enzyme can do to stress a substrate is to cause a bond to be formed between the two substrates. This is dehydration synthesis. We also call this anabolism. So you can think of the musical Annie, when the orphan Annie gets together and is adopted at the end of the movie. They join together. So if you're joining together monomers to make a larger molecule, a polymer, then this is an example of anabolism. So you see two substrates joining the active site of an enzyme. The enzyme stresses the substrates, a bond is formed, and you end up with one product. Anabolism, all of the reactions in your body that are joining, building molecules, anabolic reactions. An example of this would be protein synthesis, forming bonds between amino acids. So enzymes that do anabolism and enzymes that do catabolism, like digesting, breaking apart of bigger molecules into smaller molecules. All of those reactions, if you put them together in a group, make metabolism, which I know you've heard before. So metabolism is basically all the chemical reactions that happen inside the cells of our body and everywhere in our body. And sometimes you need more than one enzyme to create a final product. And this is called a metabolic pathway. So if you have one reactant joins an enzyme, the enzyme turns it into an intermediate product. It joins the next enzyme, becomes something different, joins the next enzyme, becomes something different, finally joins the fourth enzyme and becomes the final product. So a metabolic pathway is when you need more than one enzyme to get the final product. And a lot of times the final product will actually, when there's a lot of it in your body, it'll go back to the first enzyme and it'll shut it down. It'll say, you know what, stop working. We have enough product as it is. And that's negative feedback. That's part of homeostasis. So usually you can turn up the heat, crank up the heat to speed up a chemical reaction. And the reason that works is because of the kinetic molecular theory. If you increase the heat, molecules will bang into each other more often. Enzymes and substrates will join up more often. But of course, we can't do that inside our bodies because all the proteins will go from their nice three-dimensional shape to an uncoiled, unusable enzyme. They will unwind when they're denatured, and the active site will be the wrong shape and it won't fit, the lock won't fit the key anymore. So in our bodies, like I said before, we can't heat up living organisms to make reactions happen faster. So we had to figure out a different way to make life more feasible without making it too hot. And we do this with the energy of activation. The symbol was a big E with an A. And what a catalyst does, what an enzyme does, is it lowers the energy of activation. And the energy of activation is basically the amount of energy you need to put into a reaction to make it happen, to make it go. So what an enzyme does is it decreases the energy of activation. It makes it so you need less energy. You need to put less energy into a reaction for it to happen. So let's take a look at this animation. You see you've got the hill is representing the energy of activation. The cannon is representing the substrate trying to become the product. And if you don't have enough energy, you can't get over that hill. So it needs a lot of energy to get over that hill. That energy of activation is too high. So you don't get any products forming. On the other hand, if we look at this, you'll see the hill is lower. The energy of activation has been lowered. And the substrate can become a product because it doesn't need quite as much energy for the reaction to happen. The reason that energy of activation hill got lower is because an enzyme was added. So an enzyme will decrease the energy of activation So reactions can happen with less energy. So make sure you join me next class for all your hot questions about enzymes.